found, strangely, we didn't expect to, but we found that the glowworm uses a very similar enzyme to what fireflies use, even though they're, evolutionarily speaking, millions and millions of years apart from each other. And yet, the small molecule that they use is totally different from everything else that's been studied before. So it's quite a revelation.
There's also a practical aspect to this, because if you can make things glow using small molecular tools, you can use them in the laboratory. So you could actually ask a question of like, why do cancer cells, why are they cancerous? Or is this cell affected by an infectious disease? You can sort of interrogate the cell by finding out, is there a particular protein made or a gene made, and then have that protein interact with a glowing chemical from the glowworm. And then that chemical becomes a reporter to say, this cell is cancerous, or this cell is infected, or this cell is normal, and you can do it on a cell-by-cell basis. And it's already being done with other chemicals from other glowing creatures. But since the New Zealand chemistry seems to be different, then it could form a unique set of tools that can be used in combination with other luciferase-based research.
This next step is to prove that structure, or that it's something closely related, so by chemical synthesis of that, because then we can have enough material to do experimental work, and then understand the mechanisms, and then that can lead us through working with the enzymes, with the biochemistry as well, to making possible biotechnological tools for development. So it's a different way of probing cellular mechanisms, perhaps even something like cancer, understanding how cells are interacting or changing.
The other thing that's kind of fun is we're in a bit of a race with Australia, because Australia has a glowworm that's related to our glowworm, but they're sort of cousins, but different species. And we're in a race to identify how our glowworm works, and they're doing the same thing, and we've come to kind of different conclusions. So we find some very unique things that they haven't found yet. So we're anxious to push that ahead.